Hello everyone, welcome to the session of INI-CET November 2024 Biochemistry session. I am Dr. Rebecca James, your faculty with Marrow. So analyzing the questions, what I could understand is direct repeat. There were two questions. One was that Menkes disease question and the diabetes hormone sensitive lipoprotein lipase question. Then other questions were from high yield topics like the electron transport chain inhibitor, aneuploidy detection, recombinant DNA technology, all these derivatives of amino acids, all these were from high yield topics only. One new entry to the high yield topic which I could see is the pre-analytical error, which was not asked in any of the exams previously. So I feel that this is a new entry to the high yield topic as far as biochemistry is concerned. Then there were a few questions on the color coding of the vacutaneous, which I am not discussing in biochemistry session because that is already discussed in the pathology session. So that is actually a sharing topic between biochemistry and pathology. And most of the questions were one-liners. Few scenario-based questions were there. Okay, so let's dive into the question. First is in the current scenario, treatment for urea cycle disorder is available. Administration of phenyl butyrate can decrease ammonia formation. Which amino acid provide ammonia to phenyl butyrate? So the answer here is glutamate. As far as these options are there, the answer here is glutamate. I'll explain it. So for that, first we will see about the handling of amino group, where we see that any alpha amino acid when it has to enter into catabolic pathway, it has to discard the ammonia, but that amino group is not discarded freely. Instead of that, because of transamination, we know that this ammonia is getting concentrated as a non-toxic form, which is called as glutamate, right? Now, this glutamate is transported not as glutamate, then as what? It is as glutamine. So, the glutamate is converted to glutamine and ammonia is transported to the liver as glutamine. In the liver, what happens? The glutamine is converted to glutamate. Then what is released? Ammonia is released. And what happens to that ammonia? It enters into urea cycle. Again, the glutamate is converted to alpha ketoglutarate. The enzyme is glutamate dehydrogenase. And what happens there? Again, one more ammonia is released and it enters into urea cycle. This is how ammonia is handled carefully in our body, where it is detoxified in the liver, right? So, if there is any urea cycle disorder, this ammonia detoxification is affected. So, what are we giving? We are giving a treatment which is called as acylation therapy. So, in acylation therapy, one treatment that we give is phenyl butyrate. It is actually a prodrug which is metabolized to phenyl acetate. This phenyl acetate is going to combine with which amino acid? Glutamine, and it forms phenyl acetyl glutamine and it is excreted in urine. This is how the body or phenyl butyrate is actually scavenging the ammonia, right? Now the question is, in the option there is no glutamine. Actually, glutamine is the amino acid which is directly combining with the phenyl acetate. But in the option, there is no glutamine. Then what we should choose? We can choose glutamate also. That is why I told you all these stories. That is, when phenyl acetate is combining with the glutamine, we know that glutamine is formed from glutamate. So, in other words, the ammonia which is carried in the glutamate from the amino group of amino acid is converted to glutamine. And this glutamine is combining with phenyl acetate, means that if glutamine is not there in the option, you can also write glutamate as the answer because it is also contributing to the formation of glutamine, right? So, this is how I have chosen the answer glutamate for that question, right? Then, what is the other acylation therapy? The other acylation therapy is, instead of this, what we can use is sodium benzoate. Sodium benzoate, it is combining with the glycine. Now, this glycine, when it combines with sodium benzoate, it forms benzoyl glycine, which is otherwise called as hippuric acid. And this hippuric acid is excreted in urine. Again, by combining with the glycine, how it is going to scavenge the ammonia? It is by, so for that we should know how glycine is formed in the body. Glycine is synthesized in the body from ammonia plus 1 carbon dioxide plus 1 carbon group, which is donated by, or which is a 1 carbon group which is bound with tetrahydrofolic acid. Okay, so these three amphibolic intermediates is combining and it is becoming glycine, right? So when there is excess ammonia in the body, if we are giving sodium benzoate, these sodium benzoate will combine with the glycine. When it combines with glycine, the body is forced to synthesize more and more glycine. So when body is synthesizing more and more glycine, what will happen? These ammonia will be used up. That is the mechanism of action of sodium benzoate. Okay, so if sodium benzoate was there in the question, then you have to choose glycine. If phenyl butyrate is in the question, then we can choose glutamine. But as glutamine is not mentioned in the among the option, I have chosen glutamate as the answer, because glutamate is also indirectly contributing to glutamine, which is combining with phenyl acetate. Okay. Now we go to the next question. Which among the following are appropriate technique for chromosomal aneuploidy, in situ hybridization, conventional cytogenetics, Sanger sequencing, polymerase chain reaction? It was actually a multiple response type of question. I will discuss this and then I'll come to the answer. Okay, so which are the techniques to detect aneuploidy? The techniques to detect aneuploidy starts from the conventional technique onwards. If I am starting, I will come to this slide later. This is actually from Wiley Online Journal, where they have correctly navigated the various techniques of aneuploidy from the conventional method to the latest method. So I'll just go through that and then I'll come to the answer. So first thing is that conventional cytogenetics, that is our karyotyping, which we use, that is called as a G banding technique. You all know that. So the diagnostic accuracy of the conventional karyotyping is given as 99 to 99.4 to 99.8. It is in the amniotic fluid. If we are using a chorionic villus sampling, that is especially for prenatal screening, then it is 97.5 to 99.6, right? So that is one. That is conventional cytogenetics has now replaced with molecular cytogenetics. Molecular cytogenetics means that it is one among them is the fluorescent in situ hybridization. Okay. Now the introduction of fish, what is the advantage? Like the conventional karyotyping has a limitation, that is resolution is less. Okay, so that limitation is circumvented by this technique called as FISH. So it has evolved to be a better technique than karyotyping. Again, it is a technique to detect aneuploidy. Now we go to the next thing that is evolved is comparative genomic hybridization, which is actually a microarray based technique. So this microarray based technique, what is the advantage of this array CGH, otherwise conventional genomic hybridization? It is to reveal the imbalance across the genome, because here we are not studying a single a single gene or a particular part of a chromosome. No, here we are studying the entire genome. So in the entire genome we can find out any aneuploidy if we are using this comparative genomic hybridization. Okay, so that is the next evolved to be the better technique than FISH's comparative genomic hybridization. Again, there is the next technique which is evolved as rapid aneuploidy detection, because as far as the prenatal screening is concerned, rapid detection is very, very important, because each and every day is crucial, especially in the prenatal period. So there another technique that we are using as a rapid aneuploidy detection, which is called as RAD, is quantitative fluorescent PCR, QFPCR. So this QFPCR, the advantage is rapid aneuploidy detection, right? Now, it has several advantages over fish. One is it is less expensive than fish. Then simultaneous processing of much larger samples is possible with the QF-PCR. So, this QF-PCR has actually replaced the interface fish. So, again a better technique than fish. Then another technique is multiplex ligation dependent probe amplification, otherwise MLPA. Again, it is a technique to detect gene dosage which includes the aneuploidy as well. So, MLPA detection is again, MLPA is a technique to detect aneuploidy, but we cannot say that it's a better technique than QF-PCR or anything. We cannot say, but still we should understand that it is a technique for, it is also a technique for aneuploidy detection. So, what we have seen is there are various techniques for detecting aneuploidy, starting with the conventional cytogenetics, then molecular cytogenetics, then real-time PCR, then or QF-PCR. QF-PCR is otherwise real-time PCR. Then array CGH, multiplex ligation probe amplification. All these are techniques to detect aneuploidy. Then what about Sanger sequencing? Simple Sanger sequencing cannot detect aneuploidy because we are sequencing an oligonucleotide in a case of Sanger's, not a complete chromosome or the entire genome, because it is actually that is the technique to sequence a certain fragment of or certain sequences in the DNA. That is possible. But in the case of aneuploidy, we want to sequence the entire chromosome, which is not possible with a simple Sanger sequencing. But there is the Sanger sequencing principle or the Sanger sequencing we can use in another base, like in an NGS based platform or next generation sequencing. In next generation sequencing, which is otherwise called as massive parallel sequencing, which means at the same time different fragments are parallelly sequenced at the same time. That is called massive parallel techniques, which is NGS platform. So, if we are using the Sanger's technique to the NGS platform, then we can sequence the entire chromosome and aneuploidy detection is possible. Okay, so directly Sanger sequencing is not, but if we are using it and under this platform or massive parallel sequencing, then it is also a technique to detect aneuploidy. Now, I'll come back to this question. Which of the following are appropriate technique for chromosomal aneuploidy? I will put a tick mark to this. In situ hybridization is fluorescent in situ hybridization also we can say, because fish is a kind of in situ hybridization. Conventional cytogenetic, yes, it's a technique. Sanger sequencing, I will not say as an appropriate technique. So, I will just say plus or minus. I won't say it as a correct answer. Polymerase chain reaction, yes, because the real time, especially if the option was real time PCR, then it would have been a better option, so that I can put a tick mark. Simple PCR, as it is not mentioned conventional PCR, I am just putting a tick mark to PCR as well, because it is not a conventional PCR that can detect aneuploidy. It's a QF PCR or a real time PCR. Okay, so out of this, the appropriate techniques, correctly speaking, it is A, B and D, but I am not seeing any option called as A, B and D here. So, I am giving the answer as A, B, C, D, because when you consider these two, conventional polymerase, like as it is not mentioned whether it is what type of PCR. So, a type of PCR called as QF PCR or real-time PCR can detect aneuploidy. Okay, then Sanger sequencing, it is not mentioned whether it is simple or conventional Sanger sequencing or it is massive parallel sequencing. It is also not mentioned. So, if we can take it to NGS platform, then if the Sanger sequencing principle can be taken to NGS platform, then also we can detect aneuploidy. So, I am giving the answer as A, B, C, D. But if there was an option called as A, B and D. Here I am not seeing those options. That's why I am not giving that answer here. So here I am giving A, B, C, D. If it is mentioned like A, B, D, it was there in the option, I would choose that as the answer, not A, B, C, D, right? Because Sanger sequencing mentioned never mean that it is NGS. NGS, we have to specifically say that it is NGS because it is yet another technology which they have to say in the question, in the option, which is not mentioned. So if there was an option called as A, B, D, I will choose that as the answer. If it is not there, then I will choose A, B, C, D as the answer. So I am giving A, B, C, D as the answer with a point to note, that is, learn rather than learning what is the answer, because that is already over. Rather than learning what is the answer, you should learn what are the techniques to detect aneuploidy for further exams. Okay, so these are the techniques which I have already mentioned. So along with this, I am also saying next generation sequencing. That is also a technique where we can parallelly sequence the entire chromosome. So these are the techniques. Learn this. That is what you have to do after the exams, right? Now going to the next question, match the following. It's a very simple question. What are the appropriate precursor is what is asked. Melatonin. I usually say tyrosine, melanin. Melatonin, it is tryptophan. That I used to say in the class also. Okay, so melatonin is tryptophan. Histamine, definitely it is histidine, you all know. Norepinephrine is a catecholamine which is derived from tyrosine. Glutathione, what is glutathione? Gamma, glutamyl, cystinyl, glycine, which has cysteine. That is why we write glutathione as GSH. What is that SH? The SH is the sulfhydryl group that is present in cysteine. Okay. So the answer here is A, where you can see 1B, 2D, 3A and 4C. Okay, going to the next question. Nutritional deficiency of copper required by lysyl oxidase, which is an important key step for the formation of covalent cross-links in collagen synthesis, which gives rise to which of the following condition. Okay, similar questions were asked in the previous year as well. It's one of the high yield topic for almost every exam, NEET, INI-CET, every exam. This is a very important question. Okay, so nutritional deficiency of copper required by lysyl oxidase is an important key step for the formation of covalent cross-links, that we know. So which of the following condition can cause a condition which is almost similar to a nutritional deficiency? That is Menkes disease. Why Menkes disease? Menkes disease is due to a defect in a gene ATP7A, where a copper transporter is affected. So it is almost, it's like a nutritional deficiency, because even if we are consuming copper through our diet, it's not going into the blood compartment. So what will happen? The enzymes that require copper will be affected. One of the very important enzyme is lysyl oxidase, and lysyl oxidase is required for the covalent cross links in the quarter staggered arrangement of collagen. Okay, that's how the answer came as Menkes disease, otherwise called as kinky, steely hair syndrome. Okay, right. So going to the next question. In a patient with diabetes mellitus, the liver produces more triacylglycerol and VLDL. What is the cause for the same? Okay, so I'll come to this answer later. I will discuss this and then I'll come to the answer. So here we all know that insulin is required in the well-fed state. So in the well-fed state, what is the situation of insulin-glucagon ratio? It is very high. So if insulin-glucagon ratio is very high, we know that that is endogenous synthesis of triacylglycerol will take place, right? So this endogenously synthesized triacylglycerol is carried in nascent VLDL. This nascent VLDL is converted to mature VLDL, and later on we know that one enzyme called as lipoprotein lipase act on this mature VLDL so that it become remnant VLDL. By the action of lipoprotein lipase, fatty acid is triacylglycerol is converted to fatty acid and glycerol, and this fatty acid is deposited in the adipose tissue. Okay. So as far as this VLDL, this metabolism is concerned, what is the role of insulin here? The insulin favor lipoprotein lipase, which means it helps in the conversion of mature VLDL to remnant VLDL, so that the fatty acid is entering into this adipose tissue and it is stored in the adipose tissue as triacylglycerol. So in other words, the endogenously synthesized triacylglycerol is transported from the liver to the peripheral organs. So the second action of insulin is, when these endogenously synthesized triacylglycerol is taken into this adipose tissue, just like that, when it is giving it to the adipose tissue, just like that, it is preventing this triacylglycerol to go out of the adipose tissue as well. By which enzyme? That is by inhibiting hormone sensitive lipase. Why? Because this this triacylglycerol should hydrolyze and this fatty acid should go out of this adipose tissue only when the body is fasting. So insulin is favoring this lipoprotein lipase. At the same time, it is inhibiting this hormone sensitive lipase to keep the triacylglycerol, which is carried from the liver to the adipose tissue, inside the adipose tissue itself. Okay. So in diabetes mellitus, what happens? Either insulin deficiency or insulin resistance. So what will happen? The ulta, the reverse of these things will happen. That means the lipoprotein lipase is inhibited. So if it is inhibited, what will happen? This conversion of mature VLDL to remnant VLDL will not take place. So what will accumulate? This mature VLDL accumulate. Number one. Number two, whatever is deposited in the adipose tissue which was inhibited, like hormone sensitive lipase is inhibited by insulin, that is also not taking place. So what will happen? This inhibition is not taking place, which means this hormone sensitive lipase is activated. So if hormone sensitive lipase is activated, what will happen? This triacylglycerol, which is stored in the adipose tissue, is coming to the blood as blood free fatty acid. So what is elevated in the blood? Blood free fatty acid elevate. Along with that, this mature VLDL is also elevated. Okay, now we'll go back to that question. In a patient with diabetes, the liver produce more triacylglycerol and VLDL. Yes. What is the cause for the same? Decreased action of lipoprotein lipase and increased action of hormone sensitive lipase. Okay, so the answer here is A. Okay, so I hope that you got the concept of this answer, that is diabetes mellitus, LPL and hormone sensitive lipase. Okay, next question is a direct question from Harper. Enzyme used in recombinant DNA technology. There is a table that is given in Harper. Every time, every new edition of Harper comes, one enzyme is added to this list. Okay. In the latest edition of Harper, one enzyme that was added in this list was CRISPR-Cas9. That was asked in this exam also. Okay. So I will just show you that table which is in the Harper, which is a very important table. So there are various enzymes here. The font size here is very less. I know that. You can refer Harper for the same. So there are many enzymes given here. So I'll just show you the enzymes which was asked. One was phosphatase. Yes, that is there. Then another question, another enzyme that was asked is terminal transferase. Okay. Then another enzyme was CRISPR-Cas9. Okay. All these enzymes are used in recombinant DNA technology. Okay. Phosphatases is actually removing that 5' phosphate group, and that is for labeling this 5' phosphate group. That is, once it removes that 5' phosphate group, then we add a radio labeled phosphate instead of an ordinary phosphate. That is what we usually do. So that is phosphatase. Then terminal transferase is like sometimes this restriction endonuclease, when it is cutting this double stranded DNA, it cuts the DNA like this so that it is creating blunt ends. So we want to convert this blunt end by adding few more few more nucleotide to its end, which is called as homopolymer tailing. Okay, so for that we need this terminal transferase. Okay, then what is this CRISPR-Cas9 for? CRISPR-Cas9 is an RNA, because CRISPR-Cas9 has a guide RNA which direct this Cas9. Cas9 means it is a endonuclease. Endonuclease means it can cut the DNA from within. It is just like restriction endonuclease. That is also an endonuclease. Like that, Cas9 is also an endonuclease. So to direct this Cas9 into this double-stranded DNA, what we add with that is Cas9 is added with the CRISPR, and it has a guide RNA. This guide RNA is directing this towards the double-strand DNA. So that is why it is given RNA targeted DNA, and that is for genome editing. It is actually it is not only a research technique or a diagnostic technique. CRISPR-Cas9 is a therapeutic technique as well, because various diseases can be treated by CRISPR-Cas9. For example, sickle cell disease, beta thalassemia. There are drugs that is called Casgevy. This Casgevy is a CRISPR-Cas9 based therapeutic. So it is not only diagnostic, it is therapeutic as well. So for this recombinant DNA technology, to cut this DNA, we can use CRISPR-Cas9. So all these enzymes are required. So coming to this question, we are going to find out what is the answer for this. Enzyme used in recombinant DNA technology are isomerase, no. Phosphatase we have seen, terminal transferase we have seen, CRISPR-Cas9 yes. So the answer here is C, 2, 3 and 4. Okay. So this is a chart which is given in Harper. So please do learn all the enzymes. That means this is now a high yield topic. All the enzymes required for recombinant DNA technology, you should be learning at least the names and the primary use of it. At least these two you need to learn. Reaction, I don't think you need to learn. At least the enzyme name and the primary use of these enzymes. So going to the next question, a very simple question. Cytochrome C oxidase present in electron transport chain can be blocked by all except carbon monoxide, yes. Cyanide, yes. Nitric oxide, no. Hydrogen sulfide, yes. So cytochrome C oxidase, which is a complex 4, we know that complex 4 inhibitors are all gases. So these are all correct, but nitric oxide, no. Nitric oxide is actually a therapeutic agent. We use it for various various as a drug. We used to use this nitric oxide, and it has various functions in the body. So it cannot be an inhibitor of electron transport chain, because if it is an inhibitor of electron transport chain, then we will never survive, right? So the answer here is nitric oxide. Okay, so going to the next question, while collecting a blood sample for serum calcium, which is easily alterable, which of the following should be prevented to measure it accurately? Okay, so calcium estimation is a highly, very highly amenable for various pre-analytical errors. So we should be very careful whenever we are drawing blood for calcium estimation, because even a minute change can increase the blood. Most of these pre-analytical error is increasing the total blood calcium level. Okay. So what are the factors that affects the blood calcium estimation? I'll go to the explanation, then I'll come to the answer. So the pre-analytical errors in the serum calcium estimation in vivo, that is before drawing the blood, tourniquet use and venous occlusion, because all these two can cause venous stasis. If there is venous stasis, what happens is the water is expelled. Water is expelled from the vascular to out. So water goes out. So if that is happening, then this will increase the protein bound calcium. This will increase the protein bound calcium. So that is one. We should always prevent tourniquet use or venous. If at all we are forced to use tourniquet, what we have to do is we have to tie it just before taking the sample and immediately release. That is, maximum time is 60 seconds, right? Then the changes in posture, it can affect the blood calcium level. For example, standing. Standing is increasing the calcium level. Exercise, because exercise and fist clenching, all these have same reason. These things will increase the lactic acid. So if there is acidosis, then that is also increasing the protein bound calcium level. Okay. Then prolonged immobilization and bed rest, that is also increasing the calcium level, because prolonged immobilization causes osteoporosis. So that can definitely increase the calcium level. Then hyperventilation, that is also increasing the calcium level. So just like in vivo, there are certain in vitro errors also that can happen. The most important one is the anticoagulant. You should choose the right anticoagulant for blood calcium estimation. So for blood calcium estimation, the right anticoagulant is heparin. Again, even if I said heparin, it is not all heparin. Liquid heparin should be avoided, okay? But lyophilized heparin is advisable. And we should always prevent EDTA, citrate, oxalate, because these things can chelate the calcium level, okay? So that is why we should not use an ideal anticoagulant is heparin. But if it is given in the option like lyophilized heparin and liquid heparin, what should be the answer? Lyophilized heparin should be the answer, okay? So now we'll go back to the question. While collecting the blood sample for serum calcium, which is easily alterable, which measure should be prevented to which all measures should be prevented to measure it accurately? Tourniquet is needed. Yes, if possible, it should be prevented, right? Collecting after exercise, yes, it should be prevented. Making fist is not recommended, so it should be prevented, right? Staying immobilized for long period of time increases calcium level. It is a pre-analytical error. So as far as possible, the calcium estimation is not advisable. Like, we won't get the correct estimation if the patient is staying immobilized for long period. So that should be prevented. Then vacutainer containing citrate, I heard that that was also in the option. If that is in the option, that is also something which has to be prevented, which means all these things has to be prevented while collecting the blood sample for serum calcium, because all these are possible causes of pre-analytical errors. So answer here is 1, 2, 3 and 4. So that was all about the biochemistry question. Making mistakes simply means that you are learning better. Okay. So every time whenever we make a mistake, or if you are not very satisfied with this exam, don't worry. If you have made a mistake, just think that you are learning. Okay. So your learning process should continue on and learn from the mistake. All the best for you. Thank you.